Hello, good morning again to everyone. Thanks for joining our webinar today. So today we will talk about a topic on the qPCR, which is when you work with gene expression and you want to quantify what is the CT value. Yeah. So together with Toyobo, actually we wish to share some information on the latest technology and what is the highlight, all right, for Toyobo. So before we start, so thanks everyone again for joining us, and hopefully this session because it's Monday, right? So hopefully everyone don't have this Monday brew and start the fresh week together with Apical Scientific. Right. So just give everyone a brief highlight lah about Toyobo. Some might not know about this brand because this brand is actually a Japan-based company, which they actually quite of a innovative company, and they actually share a lot of new technology, like the KOD technology, also one of this special technology from Toyobo itself. So today we will present to you on their qPCR range, which we think this is kind of a good products to share with everyone, and also some of the knowledge about the qPCR. If everyone has any, question regarding to your project, and you wish us to assist you to help you, feel free to type in the chat box throughout the way of this webinar. So today we have here Miss Keat, our application specialist, which she will actually present to you, share the qPCR experience and information to all the experts here today. So also, last but not least, kindly share with your friends so that they won't miss any important information about the qPCR. So without further ado, wish to pass this session to Miss Keat.
Yes, thank you, Dennis. Hi, good morning, everyone. I'm Keat. I'm the field application specialist from Apical Scientific. Okay, so good morning. So I hope everyone is have already have a good rest in your weekend. Okay, so let's start on the information sharing today. Okay, so thank you for participate in this webinar with the title on harnessing the power of Toyobo ThunderBird for RT-qPCR. Okay, if you later on you have any question upon the presentation, you can just type in the chat box. I will try my best to answer you after the presentation. Okay, and if there may be some question that I cannot answer you on spot, or maybe I need some more information from you. You can also email me at support@epicalscientific.com. So as what Danny said, we can discuss later on. Okay, so not to delay, let's start the sharing. Okay, so in this presentation, I will go through on the introduction on real-time PCR. Then we will discuss on the factor that will affect your qPCR and RT-qPCR. Okay, then I will share with you on how Toyobo ThunderBird series of real-time PCR master mix can help you in your study. Also, we have another Rivertra X from Toyobo, which also can help you in your reverse transcription. Okay, so um introduction. In the introduction part, there will be quite some points which are important. Okay, so please do take note on this. Okay, so first talk about real-time PCR. Certainly, we will think of PCR, right? I believe all of you know what is PCR. PCR is the most basic application in molecular biology study. So we use it to determine multiple copy of the target gene through the amplification, right? So PCR, it involves a series of changes of temperature to generate the target amplicon. So here is the amplicon, okay? But the result can only be viewed when you finish your PCR. After you run your PCR product on a agarose gel, then only you know whether there is amplification or not. Okay, but in real-time PCR, because it's a real time, so you can actually see the increase of the PCR product in real time, okay, like this. And so here, we can call the real-time PCR, usually we will call it as qPCR. Okay, yeah. So what is the difference between PCR and qPCR? There's a little bit of different. So PCR and qPCR, basically their reaction mix will carry DNA polymerase, DNTP, reaction buffer, primer, and template. And for qPCR, there is additional of probe and the intercollecting dye. Okay, why you need these two is for your detection. Okay, so as both application also using DNA polymerase, and it works, the DNA polymerase, it works on DNA. So the template must be DNA for both application. But in real time, in qPCR, we also use cDNA as a template. Okay, so later on, I will discuss on cDNA on the next slide. And now, since PCR and qPCR are nearly similar, then why we need qPCR? Why we don't just run a PCR, right? It's a very easy things, okay? So the biggest difference between PCR and qPCR, I can say, is the application. Because PCR, you can only get to know the output after run in agarose, and agarose is not as sensitive as the signal that is generated through a machine. Okay, so you can only get to know whether there is or there is no amplification happen after your PCR is done. So that's why we call this as an endpoint amplification. And the main objective to carry out PCR is usually to get a multiple copy of the target gene for your downstream application, or to detect presence of a certain DNA region in your sample. But in qPCR, you can actually view the amplification in real time through the use of fluorescence in the reaction. Okay, and the fluorescence in the reaction, it will emit light, which will be detected as a signal by the machine. And with the amount of signal detected, so you get to know how many of the PCR product is generated from time to time. So qPCR can be used to measure the amount of the gene that is either being expressed in mRNA or it's present directly in a sample. And also with this, it can also use to detect the presence of mutation or polymorphism in a gene. Okay, yeah. Okay, so as I mentioned in the slide before, qPCR is to determine the amount of gene, either directly in a sample or a gene that is being expressed in a sample. So now what is the difference between this? Okay, just a little bit on the basic molecular. So to determine the amount of gene that is directly present in a sample means to know how many of the target gene is present in the individual organism at the genomic level. Okay, so here you want to detect the whole gene. Okay, here. Now, you can see that the gene at a genomic level, it will carry intron and exon. Okay, but a gene will need to be expressed so that it can be form a protein that will be useful to the organism. So it's just for example, like a luciferase gene in a firefly, so it will need to express to form the protein called luciferase, okay, which will categorize a reaction. Then it will result in the emission of light in the firefly, something like that. Okay, so for expression of a gene, it will need to go through transcription to copy the DNA sequence into RNA. Okay, and then the RNA, it will then undergo a alternative splicing where introns will be removed and exon can be combined in different way. Okay, so it may be combined in many different way. So the different combination of exon, it will result in different mRNA variants, which later on it will translate into different protein isomer. Okay, isoform. Okay, and with this process, a gene is expressed into a few form of protein. And for example, here, if you want to detect the amount of protein 1 here that is expressed by this gene, okay, same gene, so you need to detect the mRNA for variant 1. Okay, so if you want to detect protein 2, then you need to detect mRNA for protein variant 2. Okay, and not detect the gene itself. Okay, so this gives the two different workflow to real-time PCR. So, okay, what is the different workflow here? Okay, so to detect the gene at genomic level, you will have genomic DNA as a starting material and you will direct amplify the target DNA from there. Okay, so upon the amplification, signal will be generated, and so you can determine the amount of specific gene that is present in your sample at the genomic level. And this workflow is usually used for gene copy number study at the also the viral load quantification and mutation detection. So for example, to measure the amount of HIV in a patient blood or to detect the mutation in a gene like BRCA1 for cancer risk assessment, something like this. Okay, yeah. As another one is the RT-qPCR workflow. Okay, so um, under this workflow, there will be an additional of reverse transcription step. Okay, the reverse transcription step. Okay, it will be being performed before you can run the qPCR. So this step, it will convert the mRNA to cDNA and then continue with the qPCR reaction. So that's why we call it RT, the reverse transcription, and then continue with qPCR. Okay, this is using a total RNA as a starting material, and the reaction will target the mRNA in the total RNA. So it is usually used for gene expression study, also to detect the viral in a sample like in COVID detection, and also to quantify the mRNA that is a specific mRNA that is present in a blood sample for disease diagnosis. Okay, yeah. So now you have already know what is the two workflow. So now what gives the real-time view of the amplification in a qPCR? Okay, there are two detection methods can be used. One is through the intercollecting dye base. Okay, another one is through the hybridization probe base. Okay, so under these two methods, they have their own strength, and also it's important for you to know it before you can choose for your detection method. Okay, so the intercollecting dye base method, it is actually using a type of chemical, okay, usually is a cyanine dye, which it will not fluorescent when present freely in a solution. But this chemical, it has a high affinity to double-stranded DNA. So it will bind to all double-stranded DNA regardless the sequence. And then when it binds, it will fluorescent. Okay, so how it works is in the qPCR reaction, okay, there will be a 3-step like in a PCR. So there'll be a denaturation of the double-stranded DNA, okay, then the annealing, excuse me, of the primer to the template region, and the extension where the DNA polymerase will add the nucleotide, the DNTP, to the extending fragment that is complementary to the sequence in the template. Okay, and upon the extraction, it will means the generation of a new amplified double-stranded DNA. Okay, so the intercollecting dye, which it have a high affinity to this double-stranded DNA, it will come in and bind, and so it will give you fluorescent when it bind. And this actually means the qPCR product will increase with the cycles. So there will be more and more of the double-stranded DNA generated, and that's why there will be more dye that will be bind and give you the signal in the qPCR. Okay, so meanwhile, in the probe-based method, you will have the additional oligo, okay, which we call it probe. So this probe is a single-stranded short DNA that will be labeled with a reporter dye, which it will actually is a fluorescent dye at the 5' end, and then the quencher molecule at the 3' end. Okay, the reporter dye, it will always give signal. So when it present in a probe like this, the reporter dye and the quencher, they are in a close distance to each other. So this quencher, it will quench the signal from the reporter dye. Okay, so that's why your probe cannot be too long, because you need a certain distance for the quencher to work very well to quench the signal from the reporter dye. Okay, so upon the qPCR reaction, after the denaturation steps, okay, so the probe will come in, and then it will anneal first to the target sequence, and then the primer will anneal. Okay, so the probe will anneal first, and then the primer will anneal, and then the polymerase will come in and adding the nucleotides to the extending fragment. So upon the extending, the DNA polymerase, it will come to the region where the probe is bind. Okay, so it will cleave the probe, and this, it will release the signal, release the reporter dyes from the quencher. So now the reporter dye is free, and that's why it will give you fluorescent signal. Okay, so when the amplification go on and on, there will be more and more probe that being cleaved, and so the signal will be increased. So you get your amplification curve, okay, yeah, upon the reaction. Okay, so now when to use the intercollecting dye and when to use probe base. Intercollecting dye base is more cost effective because it's only require a forward and reverse primer and the intercollecting dye chemistry. Okay, so, and because there is only single primer set that you can use for the intercollecting dye base, so the optimization will be very much very easy. Okay, it's just like a normal PCR. So you can just adjust the temperature, so the optimization is much more easier. Okay, but this method it have some limitation because the dye can detect all the double-stranded DNA. So any non-specific binding can happen easily and affect your result accuracy. And to distinguish the specific and non-specific amplification, you will need to have a melting curve to check if there is any peak present, any primer dimer or any other unspecific peak present. Okay, so okay, now for the probe base, okay, probe base, because there is presence of the probe, so probe it will help to determine the specific detection of the target. In other words, it helps you to make sure you are amplifying the correct DNA region. So probe-based reaction, it will be more sensitive, and because probe, it will generate the signal when it bind to the correct target region. So it gives you better specificity for your qPCR. Okay, and also because you need to additional buy the probe. Okay, so the probe-based method will be more expensive. Okay, and also it can be used for multiplexing because the probe, the reporter dye, it can actually label with different fluorescent. So you can put a few set of the assay into one qPCR reaction. Okay. Okay, so I have shared about the basic of qPCR. So the basic just now basically is important for your experimental design, for you to design your experiment, so your objective, okay, and what method that you going to use. So now we want to know what is the factor that you need to consider before you start your qPCR or your RT-qPCR. And this factor, it will directly determine how smooth if is your study. Okay, yeah. So there are 5 major factors. First is the experimental design, and then your templates quality and quantity, your primer and probe, the master mix that you use, and the instrument. Okay, all this need to be considered, need to be think and plan before you start your qPCR. Okay, so for the experimental design, as I mentioned in the earlier introduction just now, so you need to first know what is your study objective. So you want to get the amount of the expressed gene, is a gene expression analysis, or you want to know the amount of the gene in the genomic level. Okay, it's a copy number analysis. Okay, and then how many gene that you going to study? Do you need to, if you have many, do you need to multiplex them? Or you can actually go for single plex that is more easier. Okay, if you multiplex, then you need to choose for probe base. Okay, otherwise, non-probe base is much more easy to optimize. Okay, yeah, so you need to plan for all this. Next is the quality and quantity of your template. I would suggest at least to run a gel for a template to check before you use for your qPCR or RT-qPCR. Okay, for gDNA, if possible, we would like to expect for a intact band, which is a high molecular weight DNA with no RNA present at the bottom of the gel, and there is no DNA stuck in the well. Okay, because of maybe presence of protein or big molecule, then your DNA will stuck in the well. Okay, and also the best is no smearing, or you can also have a less some smearing due to some DNA degradation. Okay, and then you can check for the purity of the genomic DNA at your spectrophotometer nanodrop. So usually pure DNA at adsorbent to 60280 ratio, it should be around 1.8. Okay, so you may ask me, how about if I get smearing for my gDNA? I get all smearing totally, there is no band there. Is it okay to use? You can try to use it because um, the qPCR, the fragment for qPCR is usually kind of short. Okay, so smearing or degrade gDNA will still can be used for amplification. Okay, while for RNA in agarose, you should see two bands, okay, which is for the subunit of RNA. And there is no faint smearing at the bottom of the gel, and which it should be maybe a small RNA, or sometimes it's a degraded RNA. But if there is also, it is still okay. Okay, but if there is upper, there is band at the upper, okay, above the 1 kb ladder, then it will means that is gDNA. Okay, for RT-qPCR, we don't want gDNA. Later on, I will explain it. So make sure there is no band or no gDNA observed in your RNA when you run in gel. Okay, yeah, for purity, at the adsorbent to 60280, the pure RNA should be around 2. Okay, yeah. Okay, next for primer and probe. Okay, primer and probe is very important to determine the region that you amplify, or to determine you are amplifying the correct target region. So for primer, do design it according to the general primer rules. Okay, while the probe, it should have a higher melting temperature, because as I mentioned just now, probe will bind first before your primer. Okay, yeah, so you need a higher melting temperature for your probe. And then for gene expression study, is it will be best if you can design to have exon spanning. Okay, yeah, today I won't be go very detailed into primer and probe design. If you wish to have this or learn this, you can put in the feedback form later on. So we'll see if we can plan for this. Okay, yeah. Um, Next is on master mix. Master mix, it will determine how smooth your qPCR reaction will be. So below a few points are very important. First is the enzyme activity. Okay, so you will need a polymerase that is efficient and can amplify specifically. The DNA polymerase will need to able to withstand the temperature changes in the qPCR reaction. For example, modified DNA polymerase, it can have a high amplification speed, give you a good processivity and high fidelity, then it will be suitable for a qPCR application. Okay, and all the enzyme, they will need to have reaction buffer to maintain the pH for the best enzyme activity, and also to provide the ionic condition that is required for enzyme activity. Okay, yeah, so that's why in DNA polymerase, it also need this optimum buffer condition for its reaction. Okay, meanwhile, for some master mix, especially for qPCR, they carrying stabilizer, which it can actually protect the enzyme from degradation. Because in qPCR, you want to quantify the DNA interaction, and so it is important to ensure that there is no degradation happen in the enzyme, so that you can get 100% amplification efficiency for your qPCR. Okay, yeah, something like that. And then the concentration of the DNTP, certainly it need to be in a balanced condition. Okay, if the concentration is too low, it will lead to inefficient amplification, and if it is too high, it will actually inhibit your reaction. Okay, yeah, and do get a master mix that is good in consistency and able to help you to reproduce the similar result when you repeat your experiment. Okay, yeah. So the last one is the instrument. Okay, for instrument use, you need to know which instrument that you are going to use. Maybe you want to purchase certain qPCR machine, then you go through on the manual for the machine first, and then what is in the machine. Okay, so you can decide whether you need a ROX reference die or not for that. Okay, and then what is the cost and the budget for your qPCR machine, and how many detection channel in your qPCR machine. Okay, detection channel is very important for you to determine if you're going to run a multiplex. Okay, yeah, and then you need to you need a machine for high throughput or a normal one. Okay, yeah, so and also what is the software that used by the machine. So all this you need to consider before you proceed to order anything for your qPCR or RT-qPCR. Okay, now let's see how the Thunderbird qPCR master mix can help you in your study. Toyobo ThunderBird series, it actually have two master mix here. We are going to discuss. Okay, one is the probe base, and another one is the cyber green base. Okay, both of them, they are highly sensitive in detect low copy target, and then this is very important for viral load quantification study, because usually the target viral in some sample they are quite low, so you need a something that is highly sensitive to detect low copy. Okay, also the DNA polymerase carrying hot start technology, so it help you to avoid any unspecific amplification before the reaction start. So this it means it will actually increase your specificity of the amplification and give you effective amplification. Okay, and master mix, the master mix is actually designed with PCR enhancer, which can help to reduce dispersion of PCR efficiency between target. So it is suitable for multiplexing. When you multiplex, you have different target in commonly in PCR. If you try to multiplex, there may be some suppression of some pairs of primer, so you will get different amount of PCR product from when it happened. Okay, so this it can actually reduce all these different when you multiplex, and so you get a good uh, PCR efficiency for every of your primer pair used in your multiplex. Okay. Also, the master mix, it come with individual ROX reference die, so it is compatible to all type of machine that need or don't need for the ROX as a reference. Okay, because the ROX die reference die is come in individual. Okay, with a well-optimized component in the master mix and reaction buffer, it make it stable, and so you were able to reproduce the data in every batch of your reaction. Okay, yeah, so let me show you the result comparison. Okay, so Thunderbird master mix, it can ensure you to have a lower primer dimer formation as long as the primer design, you did check the primer design. Okay, so primer dimer, it is an unintended product, something that you don't want, due to the primer anneal to each other, rather than it anneal to the target DNA template. And this, it will causing false positive signal generated, especially when you're using intergalactic dye detection method. Okay, where the fluorescent, it will bind to double stranded DNA. So when there is primer dimer, it will also bind to the primer dimer. Okay, and you will find that your melting curve, it will show an extra peak for primer dimer. So it affect your analysis later on. And because the primer which anneal to the template, it become less. So when there is primer dimer formation, it will means it will affect your amplification efficiency, that it will certainly affect your result in quantification for your gene. Okay, yeah. So Toyobo did perform some tests, where the amplification plot here will generate through the detection using cyber green method for comparison with company A and B. Okay, and the test is to target the mRNA of beta actin gene. So the cDNA is converted from a total RNA that is extract from HeLa cell, and then here you can see from the amplification uh, plot, the Thunderbird showing a clear K and distinct K curve across all the 5 dilution. So you can see there is no significant formation of primer dimer seen in the melting curve. Okay, while for company A and B, you can see the amplification probe. It shows some unspecific amplification, especially at the lower dilution. Okay, and the melting curve, you can see that is present of primer dimer here. Okay, so here we can conclude that Thunderbird Cyber Green qPCR mix, it actually demonstrate a superior performance with minimum primer dimer formation, and it makes it a more reliable choice for your qPCR assay. Okay, so Thunderbird series of master mix come with additional PCR enhancer. And how this help? Okay, here is a comparison of Thunderbird master mix with a company C on cyber green 1 assay in detect of norovirus. Okay, so from the amplification plot here, you can see that Thunderbird cyber green qPCR mix, which is represented by the red curve. Okay, so it actually showing you a more consistent amplification across the different dilution. Okay, compared to company C, which is shown in the blue curve here. Okay, while in the comparison of Thunderbird and company D on a Tagman probe based assay, to detect the GAPDH cDNA quantification. And you can see that the red curve here, it represent the Thunderbird, and then the green curve here is represent for company D. So Thunderbird, it gives you better amplification compared to company D. Okay, yeah. So from here, actually the amplification after calculate, we know that it's actually increased from 85% to 93%. Okay, so it indicate better performance by using Thunderbird. And so we can see that the PCR enhancer in ThunderBird master mix, it can contribute to the robust amplification result and ensure reliable detection and high amplification efficiency. Okay, yeah, and this is the test on the reliability and reproducibility of a Thunderbird master mix. So now this is um, to run with the Thunderbird master mix only. So there is no comparison with other brand. Okay, so here we are actually using a low serial dilution with 2 to the power of 0.5. Okay, here. So you can see from the amplification plot, Thunderbird's master mix, it can show a minimal variability between the replicates. Okay, so additionally, the amplification curve across the threshold of the red horizontal line here, okay, the threshold at a similar cycle number has further support the consistent and uh, reliable result across different dilution. Okay, and in the standard curve, okay, so you can see that the plot show a linear relationship between the CT value and the log of template concentration, and which is actually expect in a well performing qPCR assay. So this emphasize the high reproducibility and reliability of the master mix. Okay. Yeah. Next is in the RT-qPCR. So we will have a reverse transcription step. That is very important because reverse transcription, it will determine the quality and quantity of cDNA that will directly affect your qPCR result accuracy. Okay, so like the photo just now. Okay, so mRNA is actually a single-stranded RNA, and it carries the genetic information that is transcribed from DNA in a form that it can translate into protein by the ribosome. Okay, so the single-stranded nature of the mRNA, it allows it to interact with the ribosome during protein synthesis. Okay, yeah, and reverse transcription is the process of synthesize the complementary DNA from the mRNA template. So we will use the mRNA as a template and perform a reverse transcription to get a complementary cDNA. And this process is catalyzed by an enzyme called reverse transcriptase. Okay, reverse transcriptase is commonly derived from retrovirus. And so here you can see the process of the reverse transcription. The total RNA, okay, including mRNA that we want to target for our RT-qPCR, it will be isolated from a cell. Okay, then the primer, it can be either a oligo DT or a random hexamer. It will attach to the mRNA, okay, to the 3' end of the mRNA. And then this primer, it provides a starting point for the synthesis. Then the reverse transcriptase, it will come in and bind to the mRNA and primer complex and begin the synthesis of the cDNA by adding the nucleotides, the DNTP, okay, to the growing DNA chain. So the growing DNA, it has the complementary sequence to the RNA sequence here. So in the result, it is the first strand cDNA. So you will determine the first strand cDNA that will be used as a template for your qPCR. Okay. So in RT-qPCR, we want to measure the level of cDNA in order to determine the amount of the mRNA in a sample. And certainly we want to have a full conversion of the mRNA to cDNA or near to 100% conversion. So you want to expect a higher conversion rate possible, right? Yeah. So in Toyobo Revertra S qPCR RT master mix with gDNA remover, it can be a good choice for you. Okay, because this master mix, it carry high efficient reverse transcriptase with a optimum buffer condition. So this buffer, it will facilitate the enzyme activity and the reverse transcription reaction. Okay, and it can be complete in just 15 minutes, and the modified enzyme, it can reduce bias on the synthesis, and it is highly sensitive to detect low expression mRNA. Okay, and one more important thing is it is compatible to all types of qPCR master mix. So the reverse transcriptase that commonly found is the MLV reverse transcriptase. So it is derived from a monolemurine leukemia virus, okay, a virus. And this reverse transcriptase is commonly used in molecular biology to synthesize cDNA from RNA. Okay, and this reverse transcriptase, usually it consists of two main functional domain. So one is the RNA-dependent DNA polymerase domain. It is responsible for synthesis of cDNA, and it will read the RNA sequence and add the complementary DNA nucleotide to form the cDNA strand. Okay, so another domain is the RNase H domain, and this domain is responsible to degrade the RNA strand of the RNA DNA hybrid that form during the reverse transcription. So by using this transcriptase, after uh, the reverse transcription, you will find that the RNA template copy into cDNA, and the RNase H, it will actually remove the RNA strand, so you get a single-stranded cDNA. Okay, in Revertra SRT master mix from Toyobo, they have the genetically improved version of this reverse transcriptase, and this improved enzyme, it have been modified to enhance the performance of specific for the specific molecular biology application, particularly for RT-PCR and RT-qPCR. Okay, so the improved version of the reverse transcriptase, it can actually increase the thermal stability and enhance fidelity and improve the processivity of the reverse transcription. So when the processivity improve, it will give you a longer cDNA amplification. Okay, yeah. So this, it can give you a efficient and reliable cDNA synthesis. Okay, in Toyobo Revertra SQPCR RT master mix, it comes with the gDNA remover. Okay, as I mentioned just now, you don't want to have gDNA in your RNA when you want to use it for your RT-qPCR. So why this is important? Okay, here you can see that the agarose gel here, it actually showed the amplification of GAPDH cDNA under different condition involved is without or with genomic DNA removal. Okay, so here you can see that in well number 1 here, Okay, well, number one, the sample is treated with DNase 1, okay, to remove the gDNA, and then it has performed the reverse transcription to get the cDNA. So here you can see a clear band is visible. It indicates the successful amplification of the GAPDH cDNA. And since the DNase has removed the gDNA in the sample, in the RNA sample, so this band, it actually represents a true cDNA conversion. Okay, so confirm that the RT-qPCR amplification is specific to the mRNA template only. Okay, and in well 2, this is you can say is actually a negative control. So it is treated with DNase 1, but there is no reverse transcription was performed. So as expected, there is no band. Okay, yeah, because without reverse transcription, there is no cDNA generate. So after the qPCR, you won't get anything. Okay, and because the DNase one have removed the gDNA, so it confirmed that there is no DNase, no genomic DNA present. So no unspecific amplification happen. Yeah. Now, in well number 3 is a sample that was treated with uh um that is not treated with DNase, okay, but it go through a reverse transcription. Okay, so you can see a very strong band here. It indicate that is amplification. However, you can see that this band, it's actually thicker. So it actually include possible the amplification from cDNA and also from gDNA, because there is no gDNA removal is being done. Okay, so this demonstrates that there is potential of gDNA contamination in the absence of DNase treatment. Okay, so this can lead to false positive result. Okay, the last one is the one that in gel number 4. So this sample was not treated with DNase, and there is no reverse transcription being performed. So when there is no reverse transcription, it actually should have nothing. But now you can see that is banned. Okay, because there is no DNA removal happen, so there is amplification from genomic DNA. Yeah, so this show you that gDNA, it can be amplified if it is not removed in your um RNA. Okay, so it will affect the accuracy of your qPCR. And DNase 1 is a very commonly used treatment to remove genomic DNA in RNA sample. But for a DNase 1 treatment, it actually is kind of time consuming. Typically for the enzyme to remove the genomic DNA, it will already takes you around 15 to 30 minutes for the enzyme activity reaction. Okay, and follow by it, you still need to inactivate or remove the DNA, or using a cleanup to clean your RNA. Okay, so the cleanup maybe will also take you another 5 to 10 minutes. Okay, so the total duration of DNase 1 treatment, it may typically takes you around 20 to 40 minutes. Okay, but in river trial, it's the kit come with DNase 1 solution. Okay, the kit come with the DNase 1 solution, and it incorporate the gDNA removal step into the protocol. So it will take you about 5 minutes incubation at the 37 degrees Celsius to remove the gDNA, and you can proceed direct to your reverse transcription to determine your cDNA. Yeah, so it only takes you a very short duration for this process, and is incorporated into the protocol, so it is your the whole reverse transcription process. Okay. Okay, so it come to near end of my sharing. Okay, so I will summarize a little bit on the whole sharing here. So I have shared on the two workflow for reverse for the real-time PCR, which is the qPCR and the RT-qPCR. So qPCR is using genomic DNA as a template, and it directly amplified the target gene to determine the amount of the gene in the genomic level. And RT-qPCR is using the total RNA as a starting material, and it target in the amplified to detect the mRNA. So this it will require the reverse transcription, additional RT step to reverse transcript the mRNA into cDNA, a complementary DNA, and continue with the qPCR after this. So this it will help you to measure the gene that being expressed in a sample. Okay, and to make it a real-time PCR, so you can see the amplification in a real time, you can choose two different detection method. Okay, one is the intercollecting dye-based method. Another one is the probe, or you can say it's a hybridization probe-based method. Okay, for intercollecting dye method, you will need only forward and reverse primer. Okay, so it can be used for single plex reaction only. The advantage of this detection is it is easy to optimize and is very cost effective. As for probe base, you will need to have a forward and reverse primer and an additional probe that is labeled with fluorescent reporter dye and a quencher. Okay, so this probe will help you in confirm your detection of the target. Okay, but with this method, you can use it for singleplex, or also use it for multiplex of a few assay in one reaction. And with additional of probe, it will give you higher sensitivity and better specificity in the detection. Okay, yeah. And here we have Thunderbird, Cyber Green, and Probe qPCR master mix that I have shared with you just now. These two master mix can help you in your qPCR detection, and they carry enhancer that will give you a better DNA polymerase activity, okay, with the optimum reaction buffer. And the PCR enhancer, it will give you higher sensitivity, specificity, and more effective amplification. So you can have a reproducible data by using these two master mix. And meanwhile, for reverse transcription, we have Toyobo Revertra S qPCR RT master mix that come with gDNA remover. So this is very important, because this master mix is actually incorporate the gDNA removing step into the RT step. So it gives you a high efficiency and better sensitivity in your reverse transcription reaction, and the handling duration is short. Okay, yeah. So in this presentation, I can only share with you these three products. So actually under Toyobo, there is still many products for the qPCR series and RT-qPCR series, and you need cDNA synthesis kit also. Yeah, so if you are interested, do contact us because now we are going to have a promotion, right? Yeah, Dennis, we are going to have promotion for Toyobo.
Yes. Yes, so in compliance with this Malaysia Day, right, and also Merdeka Day and Malaysia Day, so we have a flash promotion for just 1 month time. So starting today onwards, after this webinar end, so we would have this promotion, a set pricing of 1,963 ringgit for 2 kits. Means you can mix and match any 2 kit from FSQ 301, which is a cDNA synthesis kit with gDNA removal, together with either you wish to look for the proof based master mix or cyber green master mix, and you will see another called QKD is actually we didn't mention here, but this is a high fidelity qPCR master mix, which is KOD based. So you can mix and match any two, and you will get a special pricing of 1,962 ringgit. And on top of it, if you are buying bulk, you mean like, later Q4 or for next year as well, you want to stock up a bit. So, and you buy any 3 set, you will free 1 set. 1 set means what? A direct combination of both. And it means you buy 3 set, you get 4 set lah. Basically, we mentioned like that lah. And you buy 5 set, you get 7 set. Something like that. So we are happy to actually share with everyone for this special promo, which only happened in 1 month, which is the Malaysian month in September. And you can start actually inquire us through cascade@apicalscientific.com, or you can contact the product specialist that you are familiar with to actually entitled for this promotion. All right. So thanks a lot for this one.